Hello and welcome to part 3, I believe it is, of microbiology weeks 3 and 4 for midterm. This is just a quick recording to review the standard plate count method with you to help you prepare for your midterm practical exam. So in the standard plate count, this is a technique that's used to enumerate or to count the number of bacteria that are found in a sample. We can't necessarily go in and look at an entire sample, a whole milliliter of sample. That would contain so many bacteria cells that it would be really difficult to count that many. So we use a method known as a tenfold serial dilution. This is a method that's used to dilute the original sample down enough that the numbers that we're going to look for are countable. Now, the method that we use in our laboratory is based on FDA standards for the dairy industry. So some of the standards and numbers that we'll be talking about in this particular recording are, again, referring to the dairy industry and the count, the microbial count for raw milk in order for that milk to be considered safe for pasteurization. All right, so in our serial dilution series, the first thing we have to do is we, of course, get our sample, and then we have to dilute that sample down using a tenfold serial dilution series. Tenfold is the best way to go because all we have to do is count zeros. It's easy to calculate, and they're easy numbers to work with. In order to carry out this method, the standard plate count method, two things that are very important that you must know is conversion factors. So you need to know that 1,000 microliters is equal to 1 milliliter, which means that 100 microliters is equal to 0.1 milliliters. We also want to make sure that we know scientific notation. Sorry about that. Scientific notation. So in scientific notation, we have, of course, 100 is equal to 10 to the 2. 1,000 is equal to 10 to the 3, right? 2 zeros, 2. 3 zeros, 3. But this also works in reverse. So if we have 0.01, this is equal to 10 to the negative 2. And 0.001 is equal, I'm getting a little too far there, to 10 to the negative 3. So you just want to make sure that you know your scientific notation and your conversions when you're ready to get started with a serial dilution series. So let's go ahead and get started now that we have that covered. Here we have our sample. Now in our sample, this sample may contain 5, 10, 15 milliliters. We're only going to be removing 1 milliliter. So this dilution series, just so you're aware, our dilutions are based on volume, not on concentration. And this is because I'm counting bacteria cells, and bacteria cells are not soluble. So they're not in concentration, they're in volume. They're in an actual microbial count. So this serial dilution I am carrying out is being carried out on volume, not on concentration. Also, when we move a sample from one tube to another, we're going to have to calculate what's called a dilution factor. So this is the dilution factor formula right here. To make it a little easier to understand, a dilution factor is calculated by taking the volume that is being moved, how much I am moving from one place to another. I'm going to multiply that by the dilution factor of what it is that I'm moving. I'm then going to divide that product by the total volume or total final volume that I make by actually moving it. So in this case, let's just set up a series here. I'm going to set up a bunch of test tubes ahead of time. Then we'll come over here and we'll just throw a little wrench in there and make it a different volume. Now, my sample is going to contain 100% of the cells that I would normally count. This means that my dilution factor, I'm not moving anything. My final volume is 1 ml, so my dilution factor is always going to be 1. The dilution factor of your sample is always 1. That is a given. Now I'm going to remove 1 milliliter from my sample, and I'm going to add that milliliter of sample into a tube containing 9 milliliters of water. When I add that, 1 plus 9 is 10. I'm going to plug these values into my dilution factor formula and calculate my dilution factor. So I take a look at part 1, volume moved. Well, this is my volume moved, so it's 1 milliliter. I'm going to multiply that by the dilution factor of what I moved. In this case, it's from my original source, which always has a dilution factor of 1. So I'm going to leave this as a 1. And I'm going to divide this by the final total volume that I made by moving it. 1 milliliter plus 9 milliliters is 10 milliliters. My milliliters cancel out, my units cancel out. 1 times 1 is 1. Divided by 10 is 0.1. This is my 10 to the negative 1 dilution. I always convert the result of my equation into scientific notation. Now I'm going to remove 1 milliliter from that dilution, and I'm going to put it into another test tube that contains 9 milliliters of water. When I do this, I need to calculate the dilution factor for my second tube. So I'm going to plug my numbers into this formula again. I'm going to do it just up here so it's a little easier to see. So the volume I'm moving is the same. It's 1 milliliter. Oops. There we go, 1 ml. I'm going to multiply this by the dilution factor of the tube that I moved. Right? So I didn't take it from my original sample. I took it from my 10 to the negative 1 dilution. So this now becomes 10 to the negative 1 instead of 1. I'm going to divide this by the volume that I make by moving it, which is 1 plus 9 is 10. Again, my units cancel. I now have 10 to the negative 1 over 10. I'm dividing this by 10 again, so that means that my exponent needs to become more negative by another 1, right? So a minus 1. So this becomes 10 to the negative 2. My dilution factor is 10 to the negative 2. So if I do this one more time here, and I move 1 ml again, this is now going to be 1 times 10 to the negative 2 over 10. This becomes 10 to the negative 3. Now let's take a look at this last one. In this last one, I'm adding the same thing. I'm taking 1 milliliter from my 10 to the negative 3, but instead of adding that milliliter to 9 mLs of water, I'm now adding it to 99 milliliters of water. This gives me a final total volume of 100. So if I have the volume moved, let's use our formula. Our volume moved is 1. And I moved it from the 10 to the negative 3 dilution. But instead of creating a volume of 10, I created 1 plus 99 is 100. Now the easiest way to do this when you're looking at it is just count the zeros and add that to your exponent. So instead of making this 10 to the negative 4, I'm now going to make it 10 to the negative 5 because it's over 2 zeros, not 1. So this becomes 10 to the negative 5. Now that I have all of my plates in a dilution series, or all of my sample throughout my 10-fold serial dilution, I'm now ready to start inoculating plates. Now when I inoculate the plates, we're using regular size Petri plates. This means I cannot inoculate a plate with an entire milliliter. That is a really huge volume when it comes to number of bacterial cells. So I'm going to, in this case, dilute my series again strictly by volume. When I plate, I'm just going to use this last tube here as an example, and then we'll look at the next page. When I plate this dilution right here, I can't plate an entire milliliter. I can only plate 1/10 of an ml. This means that my plate is only going to contain 1/10 of what I would normally count. So I have to account for that change in volume in what's referred to as the final dilution factor. Since the final dilution factor is 1/10 the volume that I started out with, right, 1 ml, then my dilution factor must go up by 1. So this becomes a 10 to the negative 6 final dilution factor. So let's take a look at the next slide and see what we have here. So here I have a 10-fold serial dilution. And you can see here, since I only plated 1/10 of a milliliter from my 10 to the negative 4 tube, the final dilution factor is 10 to the negative 5. Same thing here. I plated 1/10 of an ml from my 10 to the negative 5 tube, so my dilution factor is 10 to the negative 6. Now that I have plated my bacteria, I throw it in the incubator, I leave it in there for 24 hours. Now I come back and I'm going to count the number of colonies on each plate. This is referred to as a colony forming unit, or CFU. So CFU again means colony forming unit, and it's the assumption that every colony on the plate originated from a single bacteria cell that I put on that plate when I inoculated the plates here. Right? That my dilutions are so good that when I added just 1/10 of that dilution onto the plate and spread it around, I was able to space out single bacteria cells. And those single bacteria cells, because of the properties of binary fission, those single bacteria cells resulted in a colony. So it's referred to as a colony forming unit. We count each colony as a single bacteria cell. Now, here I have, I kind of created some numbers for you just so we knew how these would work. And in the dairy industry, a plate, when you're doing a serial dilution to check milk, the plate must contain between 30 and 300 CFU to be considered a usable or countable plate. If it's greater than 300, it's called a TNTC, or too numerous to count. And if it's less than 30, then it is too few to count. Okay, too few to count. So here, my 455, this is greater than 300, so I would label this as too numerous to count. And my plate with only 12 colonies is, of course, less than 30, so this is too few to count. My plate in the middle, at 10 to the negative 6, has exactly the number of cells right in the middle, right in between 30 and 300 that I want to count. So this plate is countable. And what I'm going to count or figure out using this number and this dilution factor is I'm going to calculate what's known as the original culture density. Now the original culture density, OCD, the formula is on the previous slide, but the OCD formula is the number of CFU that you counted divided by the final dilution factor of the plate that you counted them on. Now in this case, I have 198 CFU, and I counted them on a plate that has a dilution factor of 10 to the negative 6. In algebra, 10 to the negative 6 in the denominator of an equation, this negative exponent here, I'm going to multiply my exponent by the numerator and make my exponent positive. That's just general algebra. So I now have 198 times 10 to the 6. But my issue here is I have to report everything in proper scientific notation. So my decimal point is going to have to move over twice to the left, which means I have to add 2 to my exponent again. So this becomes 1.98 times 10 to the 8th. Now, according to the dairy industry, the farmer's milk cannot contain greater than 300,000 CFU per ml, which is what this would be, CFU per ml. Those are your units. Well, I have about 198 million CFU per milliliter, and I can't have any more than 300,000. So in this case, in the case study that's in your lab manual, this farmer would not be able to sell his milk because the milk would have too high of a microbial count for pasteurization to be considered effective. So I hope this review on the standard plate count helped kind of solidify some of these processes. Make sure to use your lab manual when looking through standard plate count. At the end of this experiment in the lab manual, there are a bunch of practice questions on standard plate count that you might find very helpful in practicing these different calculations. See you in the next one.